So I'm Alejandro Vignoni, and I will start with the webinars for modeling. Please, if you have any questions, you can write them in the chat. Jake will be there taking care of that, and after there will be a few slots after I start. After I stop presenting each topic, there will be a time for questions, and then at the end we can, if anything got an answer, we can answer that at the end. Okay, good. So there will be 3 parts today. In the beginning of the webinar, the first section will be ODEs, the law of mass action kinetics, and the central dogma, and then we will, in the second part, we will go to the derivation of the Hill function from the law of mass action to know where is it coming. And then if we have time, we will have a short exploration of the Hill functions, changing parameters, and see how that affects the shape of the curve. Okay, great. So if you were in the first webinar we gave on the opening weekend, we talked about models and why we need to model in synthetic biology. So we start here with the types of model we have, and in these webinars of modeling, we will deal with these 3 that I have here. Starting from the biochemical reactions, we will get the mathematical model of a system of ODEs, and then we will reduce them to have a more easy to deal with and to relate with data, smaller model. Okay. So, but first, just in case, for the people who doesn't remember, what is an ODE, an ordinary differential equation? These are equations that have variables and their derivatives. For example, the easy one, y equal f of t. Of course, it can be more complicated. And the derivative, here I'm putting the 2 notations that we generally use. This one is the Newton notation, and this one is the Leibniz notation. When we do models, in general, the derivatives will be always respect to time, so we will use this guy, which is much simpler to write. And the definition here with the limits of the function, okay? But they, depending on how difficult they are, they can be very challenging and difficult to solve. So generally there are 2 options to deal with this. Analytically, solving for the unknown, but that again can be very hard. And numerically, in an approximate way, which is called the easier one is the Euler approximation, which is this one down here, that we will solve with a computer software. Okay. Why do we use them? Because differential equations describe biological behavior, physical laws, human activities, and much more. Here, there is a very small example of, for example, the biomass or the cell culture in a culture, cells in a culture, sorry. And the rate of change of this variable x depends on the growth rate and on the maximal capacity of the system. And when we solve this equation, we get this nice curve here, and that's it. So when we have a set of differential equations that describe a system or a phenomenon, that is what is known as an ODE model that we will talk a lot about after here. A short note on what kind of software is there to solve ordinary differential equations. There is MATLAB, and so it's the one that we will be focusing for some reasons. One is iGEMers, you will get free license for being iGEMers. And the measurement committee also has some software that you can use for calibration, validation, and data analysis of flow cytometry and plate reader data. So it's that there. But of course, there are many other softwares that you can use. Here is just a very short list in some of the explanations, but we will not focus on this more. If you have any question, then we can answer that. Okay. Okay, so let's start the first part of this journey from the biochemical reactions to the ODE, the mathematical model. At first, a small reminder, some of you may remember, but just in case, the law of mass action and the kinetic equations of chemical reactions. In this example, I have the reaction of water, which is known for many people. On this side, we have the reactants, hydrogen and oxygen. The arrow tells us that the reaction is going in that direction, and the product of this reaction is water, which we have on this side. So again, reactants, products, and this reaction is happening at the speed which is dictated by the reaction rate, which is what we put on top of the arrow here, and it's symbolized by K in this case. And then another thing important here is the stoichiometric coefficients, which are the quantities of molecules that we need for this reaction to happen. In this case, as we all know, we need two hydrogen 2 molecules, one oxygen, and with that we get 2 molecules of water. Okay, so let's start with the differential equation that tells us how hydrogen 2 is changing. Remember, the dot on top is telling us derivative with respect to time. So the rate of change of this molecule, of the concentration of hydrogen, will have some terms coming from the reaction, and we will learn or remember how to get them. So first, this rate is proportional to the product of the concentrations of all the reactants. And in this case, we have 2 times hydrogen and oxygen. So it's hydrogen, hydrogen, oxygen, which give us this term, okay? And then the rate is proportional to the reaction rate, which tells us the speed at what this reaction is happening. And these 2 is coming from the stoichiometric coefficient of the molecule that we are dealing with, in this case, hydrogen 2, which is telling us that at the time that one reaction occurs, 2 molecules of hydrogen 2 will disappear, let's say, to make the water molecule, okay? So these 2 is coming from here. And then we have the minus sign here, which is telling us that hydrogen will be decreasing because it's being converted converted into the water molecule here, okay? Okay, that's fairly easy. So if we go to oxygen and you look at the equation, how oxygen is changing, it has the same shape, the same form the previous one. Again, the product of the concentrations of the reactants, hydrogen, hydrogen, that's why the square is here, because we have 2 hydrogens there, and oxygen, the rate, the speed at what the reaction is happening, and then here there is nothing or a 1, because every time that the reaction happens, 1 molecule of oxygen 2 is being used, let's say, to get us the water, okay? And again, the minus sign is because the oxygen is disappearing, let's say, to become the water molecule. Very good. Oh, there is a typo here that should say oxygen, sorry. And now, if we look at the water, and we want to know how it will be changing. Again, if you look, these terms, they look the same in the 3 equations because the reactants are always hydrogen and oxygen, hydrogen, hydrogen, and oxygen. And now here, difference with the ones before, here there is a plus because the amount of water molecules is increasing when the reaction happens. And the number 2 is because every time one reaction happens, we will get 2 molecules of water, the stoichiometric coefficient here. And again, the rate of change, the rate of this reaction happening, K, which goes here. Okay. So if we put them all together, we have the three equations that give us the dynamics of the system, the kinetics, and they will tell us how, if we put oxygen and hydrogen in some container, let's say, we put them together, this equations will tell us how they will change in time. So I don't know if you can see the video properly in the Git of the modeling webinars. You can get the video alone to watch it later, but I will tell you what we try to see here. On top, there is the graph, which is changing in time, telling us the amount of molecules of, sorry, the amount of molecules of oxygen in red, hydrogen in gray, and water in blue. As you can see, oxygen and hydrogen are going down as we are getting more and more molecules of water. And here is a very small animation that is telling us how this conversion is happening and why we have less molecules of oxygen and hydrogen at the time when they become the water molecule, okay? Just to get the idea of what these equations are telling us. What we have to do to solve this system of equations is we have to know K, and we have to start with an initial condition is called of how many molecules of hydrogen and oxygen we have, in this case 20 and I don't remember, and that will lead us to have the solution of this system, okay? So as a summary, I have the graph here again, already plotted, and this, as you can see, this tells us at each instant of time how many molecules we will have of the three elements that are here in the reaction, okay? So the very small summary that to know what we were doing, we will get one equation per species. So in this case, we have three, hydrogen, oxygen, and water. We will have three differential equations, these three. And the rate of change depends on the stoichiometric coefficients of the species times the reaction rate and the product of the concentrations of the reactant, as we saw before. We will use this to model the central dogma, which is what is interesting for us, how DNA genes are converted into proteins and how this is happening dynamically and how is that changing in time, okay? So very short reminder of the central dogma. We have DNA, the genes, polymerase, RNA polymerase goes there and transcribes the DNA, and we get the messenger RNA, and then the ribosome translates into protein. Very, very simplified way of saying what is happening there in the cell, okay? So if we write an equation, a reaction to describe these processes, we can write this, where we have, again, the transcription process, the translation, and also these other purple arrows down here are telling us that these molecules are degrading. So RNA is degrading at the rate D1, and protein is also degrading at the rate D2, which is also including the let's say, the dilution related with the growth, because the proteins that we will use in general are very stable, so they are getting diluted with growth. With DNA, they are much faster. The degradation rate is much faster, so it's not necessary to do that, okay? We will talk later. And then we have the rates here, K1 and K2, that tell us at the speed that these reactions happen, okay? So let's try to write the equations for this set of reactions. If you look, we don't have only one reactions. We have one here, another one here, and another one, the degradation of the protein, okay? So let's focus on the first part. Gene gets transcribed into mRNA, and this, what happens is we will get a positive term in the derivative of RNA. Remember, the dot here is telling us derivative of the amount of mRNA in respect to time. How is it changing? And we will get mRNA will be getting created, let's say, at a rate which is proportional to K1, the transcription rate, and the amount of genes we have. I didn't say it before, but we are talking first about constitutive gene expression. So this is happening all the time. The gene is always on, let's say, and it's getting transcribed constitutively. Then what else is happening with the mRNA? It's getting degraded, sorry, which is happening at a rate d1. And the term that goes in the equation is, as we saw from the law of mass action, is the concentration of mRNA times d1, okay? This term. And the one before was the amount of genes we have times the rate. So we are following the rule to construct the differential equation here, okay? So now we go to translation, and now we want to see how the protein changes in time, the amount of protein. So we will have, from this reaction, we will have that there is a positive term, which is K2 times the amount of mRNA we have, this one times this one. And then we will have a negative term, which is referring to the degradation and dilution of the protein, which is D2 times the amount of the protein we have here, okay? So with that, we get the full set of two differential equations that tell us how mRNA and protein will change in time, depending on the value of the parameters, okay? So, sorry, if someone is with the mic open, can you just mute it so we don't hear any, if you want to ask a question later, you can open your mic, but to don't disturb, better to have it muted, okay? Thank you. Okay, so we have the equations here, and okay, so we have it here. Let me show first a small simulation of this. And we have the mRNA, the protein. We have to write the model first, the differential equations, which are written here. And then we have to give values for the parameters. This simulation is done in MATLAB. And then a few configuration lines of how much time we need and tolerances, initial conditions. And with this command, we solve the system of equations and we get the solution that we can plot. I will not be spending much time here. One important thing here is a link to a very interesting discussion and explanation on why there are different types of ODEs, stiff ODEs, different types of solvers. Here we're using ODE 23T, which is for stiff systems that we normally get with when we do systems of reactions. And also there will be a webinar from MathWorks in July, which will be focusing on how solving differential equations and much more using MATLAB. So take note of that in your calendars and go there if you like. Okay. So what we get when we do the simulation is how changes in time the amount of protein, which is in yellow here, and the amount of mRNA. And in blue, we have the amount of genes we have, which we said here in the parameters that we had 17. For example, this kind of origin of replication has that number of plasmid copy numbers. So that one will be constant. And then the expression of protein will happen with this shape in time given by the differential equation, okay? So just final remarks of this part. From the reaction that we had here, we got the equations, and with these equations, we can get a simulation. Some small things. As I said before, the amount of genes is considered constant, and it will depend on the plasmid that we are using. But they are generally known values of how much copies of the plasmid are given by a origin of replication. And small things, RNA and ribosome, RNA polymerase and ribosomes are considered sufficient enough, so they are not limiting in this process. It's a general assumption. And then the This we'll see later, but binding and unbinding processes are much faster than transcription and translation, and the protein degradation includes the growth associated with dilution, as we said before, okay? So this is the end of the first part. If we have any questions, okay, so sorry about the mic thing. It was one of us who had it open. That was a mistake from my part. If we have any Q&A, no questions. We can have a small break if you like. Jake, I don't know what you think. We are already ahead of.
You've got 2 questions in the Q&A to answer.
Yes.
One is, why don't you answer those and then go forward?
Okay, thank you. Sorry, I was trying to look at them. Okay, I have more. Okay, so binding and unbinding we will see on the next part, but I was referring to the part of this process where, for example, the polymerase goes to the promoter, to the operator in the promoter and binds and start transcription. That the binding is the process where 2 elements get together, and we will deal with that in the next one. Okay. What about ribosome loading? So this assumption is to deal with that. We are assuming that our gene is not putting too much burden into the cell, so there are enough ribosomes for the cell to do what it wants to do, and also to produce our protein that we are putting there. There are models. People, actually, that's an open question in research. People, myself among others, are working on trying to model how the cells is distributing the ribosomes it has to deal with what it has to do and to produce the protein that we want. So in this case, we are not taking that into account, but people is trying to answer that question with models that are, of course, much bigger and more difficult to deal with. The next one, I hope I answered that one. How do we predict the amount of protein produced through promoter activity? That's the topic of the next part. We will use the Hill function that we will derive first to understand how the amount of protein will change depending on the promoter activity. When it's an inducible promoter, okay? And in the case of a constitutive promoter, the promoter will be modeled as the constant K1 that we put there, which is the rate of the reaction of transcription. And the last question, sorry, I was not repeating the questions. That was a mistake by me. I will do it now. So there is one question that says, I don't fully understand how the amount of gene stays constant. Surely in the model shown, it should decay as it being transcribed. So what we are saying, the gene, while it's being transcribed, it's still there. And when the polymerase goes out, the gene is still there. And in the process of duplication of the cell, there is the replication of the DNA, which ensures that in the daughter cells, we have the same amount of genetic material. Of course, there are stochastics involved there, but in general, we can assume that the genetic material from the mother goes to both after the division goes to each of the cells in the same amount. Okay. Wow, we have a lot of questions. Okay, so
I can keep answering questions in the Q&A while you go on the second part if you want. So
okay,
do a couple more, but I'll keep doing it in parallel.
Okay, here there is one which is interesting. Would be the limiting rate of RNA polymerase binding be significant if the target gene is in a chromosome versus a plasmid? that's something to consider, yes, because when we have plasmids, the amount of plasmids that we can have if we have a high copy plasmid, it could be very high. So that will make us in more need of polymerase to deal with that. So that could be something to take into account. Yes, it's an interesting question. I will answer the last two and we will go on, and then Jake, we will follow up with that. So the one question says, so in constitutive promoters, the promoter activity is equal to the rate of production. It's a part of the rate of production, because you have to, if I can go back to the reaction here, the part which is given by K1 is the promoter activity, let's say. And then we have also the translation process which converts the mRNA into protein, which also will be part of the amount. The amount of protein will be a function, let's say, of both the promoter activity that you are saying here and the ribosomal activity, which depends on the RBS that we put there and so on. And the last one I will answer here, the proteins with degradation tag by how much the half-life is decreased, such as in the case of SSAR tag. So that's also an interesting question. It's a good point that with the different types of tags, SSAR is one of the LDA tags that you can have there. You can change the value of this guy. D2 is depending on that, but that will depend of many things that will happen in the cell. So you can, something that you will have to do is to make an experiment and to measure, maybe have the protein with the tag and without the tag, and to compare the speed of growing of these curves, and you will be able to measure that difference there, okay? And the last one, very last, how does promoter strength affects the rate of mRNA formation? That's exactly the K1 that we are putting here is having together the effects of the promoter activity and the binding affinity of the polymerase to the promoter and many other elements there, but one of those is affecting them. If the promoter is stronger, the value of K1 will be bigger, and the amount of mRNA produced per gene, which is the orange one here, will be higher for the same amount of genes if we have a K1 which is higher, okay? And this one I'm not answering, the last one. It says, if transcription rates are not found in the literature, how do we find them? Very good question. We will try to answer that in the webinars from now to the end of the summer. The way you do it is you have to relate the model with experimental data. Jake later on in July will give us seminars on how to calibrate and use plate readers and flow cytometers to get good data. That, together with the models, will be the way to trying to find values for these parameters for our constructions in the lab, okay? But so in the literature, you will have some options to find that. Bio numbers is one. I will put the link later, where you can find some values of this, okay? Okay. Very good. Let's change to the next part. Second one.